Okay, so today we'll be talking about gene transcription and RNA modification. First, we're going to really talk about the overview of how DNA and proteins are made from and how RNA is made from DNA. So the first important thing to know is what a gene is, and a gene is defined as a segment of DNA that can be used to make a functional product. And this can either be an RNA molecule or a polypeptide. And I guess the most central thing to know of transcription or how translation works together is the central dogma of genetics. And this was made by Francis Crick in 1958. And it's actually how the information in DNA can be used to make mRNA and then can be translated into a protein. So there's a few steps for this, and it starts out with DNA. And then the first step it goes through is transcription. And that's basically using DNA as a script to make mRNA, which is important because mRNA can be used to make a polypeptide, which is eventually turned into a protein, which is what's in our bodies and what helps us function. So as far as transcription goes, there is both DNA and mRNA parts to it. And so the DNA has promoters and terminators, and they act kind of in a similar way as start codons and stop codons, and that's why we set up this slide this way. And so the promoters, they are sites for RNA polymerase to bind, and it signals transcription, whereas the terminator, it is what it sounds like. It ends that process. It tells it to stop. And what's also important is some regulatory sequences, and these just kind of control the rate of the transcription in the DNA. However, in the mRNA, there are start codons and stop codons. And the start codon tells the first amino acid in the polypeptide sequence, whereas the stop codon is the end of the polypeptide sequence. And there can be more than one polypeptide in each protein. These codons are just 3 nucleotide bases that basically tell the mRNA when to start and stop making the polypeptide. So after talking about specifically the initiation step, we're going to talk about how there are also transcription in bacteria, because this is just mainly the overview. The bacteria is slightly different. So in the transcription of bacteria, there are 2 important factors. And it really just has to do with the negative 35 and 10 sites, and also what's known as this 10 box right here. That's known as the Primnell box. Kind of a fun fact, named after the originator, the founder, whoever found it. And these are used to initiate the transcription. And these are all labeled negative before the promoter region, and the promoter site on that is the positive one, as indicated kind of like right here. And then there's no 0 nucleotides. There's only the positive and negative. So if you're numbering the sequence, you're going to skip the 0 mark on there. These are just basically the really quick rundown on how it works. Pretty simple. We just make, after the promoter has bound to the negative 35 and negative 10 region, the sigma factor is released and the core enzyme is able to move along the DNA to make RNA. And then it's basically just self-explanatory. It removes, and then they're both separated from each other. Next we have elongation. And some key points to remember is that the RNA polymerase slides along the DNA, creating an open complex as it moves. And another important thing to know is even though that the RNA reads to the 3-5 direction, it's synthesized in the opposite 5-3 direction. So that means even though it's reading one way, it's actually being constructed in the opposite way, or the 5-3 way. Next we have the termination step of the bacteria, and there's only 2 different ways, and they're pretty simple because they are the opposite of each other. So even though both have the formation of a stem loop. The row independent is exactly what it sounds. It does not require the row protein, but it does have a urso-rich 3 end at the RNA, whereas the row dependent site has the row protein and has the termination site where the stem loop forms. While we discussed the transcription in bacteria, we also have transcription in eukaryotes, and this is a little different. It starts out by using 3 different types of RNA, and the first type of RNA transcribes all genes for ribosomal RNA or rRNA, and then RNA2 transcribes all protein encoding genes, and then the third kind transcribes all tRNAs as well as the 5S RNA that is not transcribed by the first one. The first one does all of them except that last 5 one that the third does. We also have 2 important things involved in the initiation step, and that is the core promoter and the TAT box. And there's also things called enhancers and silencers, and the enhancers are needed to stimulate that transcription, whereas the silencers are needed to kind of end that transcription. We also have some general transcription factors, but those aren't as important. There's too many to go over each one right now, but we will talk about the termination and the allosteric versus the torpedo model. So the allosteric model and the torpedo model are very similar to each other, except that the exon nuclease kind of breaks up the other section of the RNA in the termination process. Now that we've talked about the transcription of bacteria and transcription in eukaryotes and how RNA is formed, we can talk about all the modifications that can happen in that RNA after it's made. So we've talked about how the RNA is made from the DNA, but we can also, there's many things that can happen after the RNA is made. So the first one, there's 6, and the first one is processing, and that's known as the cleavage of the large RNA transcript into smaller pieces. It's just the smaller pieces become the functional RNA molecule, and it can occur in both prokaryotes and eukaryotes. The second one we'll talk about is splicing, and that's this one right here. And splicing involves the cleavage and joining of RNA molecules. It removes the introns in here, and then from each one, so basically it's just bringing together 2 extrons. And then we have the 5 capping, and the cap plays a role in splicing the exons, the exit of mRNA from the nucleus, and the binding of mRNA to the ribosome. Then we have the 3 polyadenylation, and this is just an attachment of a string of adenine containing nucleotides to the 3 end of the mRNA. The last 2 are RNA editing and base modification, and the RNA usually occurs in eukaryotes, and it's just a change in base sequence of the RNA after it's been transcribed. And the last one is the base modification, and that's exactly what it sounds like. It just is a change in base. In the interview, we have talked about, today we have talked about how RNA is transcribed, not translated, transcribed from DNA in bacteria, eukaryotes, and then we briefly discussed all the things that can happen after the RNA has been created or formed.